Neurology, Physiology, and Anatomy, the neuron action potential. All right, so we're just going to cover the action potential of neurons as well as a couple other things for the physiology and anatomy aspects before we move on to sleep cycles. So first and foremost, remember action potentials is what's going to be leading to all of our communication within the central nervous system, and it's done by the neurons, right? So we have a normal resting potential, right? And the resting potential could be just like a negative number. And what happens is, typical for all of our action potentials, right? When we have a opening of the voltage-gated sodium channels, it leads to sodium entering the cell, causing the action potential to occur, okay? And then after the action potential occurs, the sodium channel is closed, the potassium channel is opened, and potassium flows out of the cell, okay? Leading to, right, we have increase in potassium permeability, okay? And it leads to a drop back in potential, and it can cause a drop further than the resting potential, okay? Very easy to understand. Now, the one thing that they like to ask about with the action potential, right? It's not that high yield because this is very easy, but I mean, it's high yield just for your understanding of what's going on. But a pathology they like to ask about specifically is a toxin. So there's a toxin that comes from a puffer fish. And they love to ask the question of a patient comes in, he just was visiting Japan, or he's a Japanese resident and he was just eating fish or eating an exotic meal at a restaurant, okay? They might not tell you it straight up, but puffer fish is an exotic meal in Japan that they like to eat. But the issue is that puffer fish contains a toxin, okay? And it's known as TTX. And that toxin blocks the upstroke action potential within the nervous system, okay? So you can't actually trigger the action potential because what TTX does is that it blocks stage 2, essentially blocking the opening of voltage-gated sodium channels, okay? And so that leads to the problem of not having an action potential made, okay? So that's how they will normally present the information to you. Next is our peripheral nerve, okay? Now we're going to talk about anatomy here. So this is how all of our peripheral nerves are made, and it's very easy. And I think that you're going to see a similarity to nerves and muscles because, once again, the human body sees a good mechanism of creating something, and it's going to repeat itself. So individual nerve fibers, okay? So one specific, essentially myelinated nerve fiber is going to be surrounded by endoneurium, okay? And the endoneurium, what they want you to know about this specific area is that when it becomes inflamed or there's inflammatory infiltration of the endoneurium, that is likely Guillain-Barré syndrome. That's likely how Guillain-Barré syndrome affects the nerve fibers, okay? So once again, the endoneurium surrounds one specific myelinated nerve fiber. And then multiple nerve fibers come together to form a fascicle. And a fascicle is surrounded by the perineurium. Lastly, multiple fascicles are clumped together to form the nerve, and the entire nerve is surrounded by the epineurium, okay? Although not that high yield, I will say the one thing that you should note is that the endoneurium, if there's inflammatory infiltrate within that area of the endoneurium, that is likely due to Guillain-Barré syndrome, okay? And that's how Guillain-Barré's pathophysiology occurs. Next is chromatolysis. Chromatolysis is very high yield when it comes to understanding damage within the nervous system. So what happens, or let me just first define what chromatolysis is. It's just the reaction of the neural cell bodies to axonal injury. So what happens to our neurons when they get injured, okay? So it's characterized by 3 things, and these 3 things are very important. The first thing you're going to see is round cellular swelling, okay? The cell is going to swell, and that's indicative of any type of cell damage, right? Not only neurons, but all cells will start to swell when damaged, okay? So this is no different. So you're going to see round cellular swelling, okay? Next, you're going to see displacement of the nucleus to the periphery. So the nucleus is going to go off to the side, the periphery of the cell, and the last thing you're going to see is dispersion of the Nissl substance throughout the cytoplasm. They want you to know this 3 steps or 3 characteristics of chromatolysis, okay? Now, they might say chromatolysis is also known as axonal reaction. Don't get it rattled. It's the same thing, okay? It just means that you have neuron damage, okay? It's chromatolysis is also known as the axonal reaction, okay? Now, what happens is, after this whole entire thing takes place, you can have the axonal reaction take place, and we have damage to the neuron. We have this thing known as Wallerian degeneration. This is what's going to occur after those three characteristics show up, right? So after round cellular swelling occurs, after we have the displacement of the nucleus to the periphery, and then we have the dispersion of the Nissl substance, right? We're going to see Wallerian degeneration, okay? Now, what does that mean? Essentially, all of the neuron that is distal to the injury will degenerate. Okay? And macrophages will come and remove that debris. Everything that's proximal to the injury, the axon will retract and the cell body will sprout essentially new dendrocytes, okay? That will help with communication and grow towards other neurons later on. Okay, so what I want you to take away from chromatolysis, or once again, axonal reaction, is that it's characterized by three things. After injury, it's going to have round cellular swelling, displacement of the nucleus to the periphery, and dispersion of the Nissl substance throughout the cytoplasm. So that's what happens at the cell body when a neuron is damaged, okay? And then, distal to injury, you're going to see Wallerian degeneration. So myelin debris starts to form. All of the axon becomes debris, the myelin becomes debris, and then the microglia come up and eat it up, okay? And they clear out all of this, okay? So very, very important to understand that. They could ask a lot of questions. And lastly, what happens to the proximal part is that the axon is going to retract back to the cell body, and it's going to sprout out different dendrites in order to have communication with different neurons, okay?